This abstract looked at circulating cell-free DNA and aberrations in the PTEN, PI3 kinase pathway in men with metastatic castration-resistant prostate cancer. So circulating cell-free DNA or cfDNA has really emerged now as a very, more than just a promising, but actually a very useful tool in profiling, molecular profiling of many cancers, including prostate cancer. And in fact, it's very attractive in prostate cancer because in metastatic prostate cancer, most disease is found in bone or in deep intra-abdominal lymph nodes, and it's not easy to stick needles into bone or to deep lymph nodes and get tissue back. It's painful for patients, and sometimes you don't get any information back. So cell-free DNA can be picked up in blood from a blood draw, from plasma. And in fact, with modern technology, we can find molecular alterations in the vast majority of patients. So in this particular study, we had a large cohort across both US and Australian centres, over 200 patients who had blood taken before they started treatment with standard of care agents such as abiraterone, enzalutamide, docetaxel, or cabazitaxel. And what we saw was there were three key findings from the study. We were able to robustly identify PTEN loss. So PTEN is a tumor suppressor gene which is commonly lost in prostate cancer, but it's very hard to detect loss of PTEN in blood. And the assay that we've been involved in developing can robustly identify that. And that's really important because AKT inhibitors, an AKT inhibitor in the ipatasertib in the iPotential study was recently shown to be to prolong radiographic progression-free survival when combined with abiraterone in PTEN deplete metastatic castration-resistant prostate cancer. So being able to pick it up in blood is really important. We were also able to identify, in addition to PTEN loss, another 20% of patients had other alterations in their plasma cell-free DNA that could sensitize them to AKT inhibitors or other similar pathway inhibitors because they had other alterations in the PI3 kinase PTEN AKT pathway. And the third finding from the study was that we identified men who had composite alterations or aberrations in the PI3 kinase and PTEN pathway and then the androgen receptor pathway. And we found a novel group of patients with three copy number changes in genes in these pathways who had very poor outcomes on standard of care therapies. And we think those men are the ones who could really benefit from the combination of an AKT inhibitor and AR pathway inhibitor. And so this work really, I think, has very strong clinical relevant, translational and clinical relevance given the results of the iPotential study and the fact that there are other PI3 kinase inhibitor studies coming through. And I think that cell-free DNA can really help us use these drugs better and understand and understand which patients are most likely to respond or not respond to treatment.